Okay, lecture 3, microbiology students. And so the goal here, as you can see, is to understand how we can start to learn to classify and identify microorganisms, most specifically bacteria. And so if you think about if you have a patient that comes in who's really sick with could be a wound infection, it could be a blood infection, could be a urinary tract infection, whatever it is. One of the goals is to obviously treat that infection, but one way to do that, or one thing that may be necessary to do that, is to figure out, well, what microorganism is actually causing the problem. And so to be able to identify what bacterial species it is, we have to understand what we can look at and what information we can gather from microorganisms that can be useful and informative to drive that decision-making process. And so that's kind of the goal of this lecture, is to introduce how we can classify bacteria and say what groups they belong to. And so most of you have heard of, oh, that's a staph infection or that's a strep infection. And so those represent groups or classification units. And so then once we have bacteria in these groups, what are the common characteristics that enable us to say, oh, this patient has a staph infection, but this one has a strep infection? And so that's kind of the goal of this lecture, is to introduce those topics. This idea of classification is also rooted in history of science. And so there's one individual, and so his name is right here. His last name is Linnaeus. And so he was responsible for introducing much of the system of classification and identification that we use today. And so there's a term we use to describe this. It's called taxonomy. And you can think of taxonomy as three pieces to a complete puzzle. And so one is, if you were in Linnaeus' shoes, his goal was to try to organize and put all living things into groups. And so that's what we call classification. So what are going to be meaningful groups? So he came up with his system. It's been modified a little bit, and we'll talk about that, but that's what we mean by classification, putting things in useful, meaningful groups. Nomenclature is a fancy word for naming. And so not only did we want to have groups, but we wanted to have common shared names that all individuals kind of across the planet would understand, so that you could have a useful conversation about something. And so, for example, when we use the term animal, that's obviously different from plants. And so those are both groups or classification units that have meaningful names or nomenclature. And then identification is when a new organism is discovered. We can then use that existing set of classification groups and names to put that into that newly found organism into the system. And so this is really important for organizing this vast sea of information of all living things. And so Linnaeus really established the groundwork of this system. And some of the things that still remain even today is what we call a binomial nomenclature. And so this is two names. And so the two names that we're talking about is every living thing on the planet, once we have it identified, has a two name binomial nomenclature. And so the two names are the genus and the species. And so that's the most descriptive we can be. So for example, I mentioned a staph infection. There's multiple species of Staphylococcus. And so most of the time staph infections are going to be the genus Staphylococcus, but the species aureus, so Staphylococcus aureus. When we talk about strep infections, there's a variety of strep infections. Which specific strep is it? Is it Streptococcus pyogenes? That'd be genus species. Is it Streptococcus pneumoniae? That's a genus species. And so we can do that for bacteria. We can do that for animals. So humans are Homo sapiens. And so this system of a binomial nomenclature is attributed to Linnaeus. And he also came up with that genus species gets really specific. So which exact organism is it? But he did it in groups. So if you don't take all living things and just put them down into genus species right away, but you break them into more manageable groups. So this is the system he came up with that's still in use today. So kingdom, you can take all living things and divide them into kingdoms. When Linnaeus did this, he only recognized plants and animals. So you had two kingdoms. We now know that we can break them down further, and so it's evolved a little bit, but this hierarchy of groups still exists today. And so its kingdom is the highest, and then you can take all kingdoms and break them down into what are called phylums, or phyla, plural, phylum, singular, and then phylum can be split into class, and class into order, and family, and then genus, species. And so just to illustrate this next slide before we get there, we'll look at that organization in just a little bit. I mentioned Linnaeus had only 2 kingdoms, animals and plants. More recently, you can see that, again, the dates aren't overly important for ours, but just to see the time scale of it. Whitaker, another scientist, proposed the systems of kingdoms that we kind of use today. And so not only are there animals and plants, but we also recognize that there's an entire kingdom that is all the fungal organisms. So fungus is its own kingdom. And then prokaryotes, sometimes this is called Monera, but prokaryotes. And then Protista is not the best kingdom, but that's what it was called. And basically that included all eukaryotes that did not include animals, plants, or fungus. And so this has things like protozoan and algae in it. So this is kind of a hodgepodge of different things. So those are what Whitaker proposed. It has been modified a little bit. We'll talk about that in just a second. But before we do that, we can talk about this different, at least see an example of this hierarchy of classification that Linnaeus came up with. So Linnaeus's classification starts with kingdom and then phylum. And so I'm going to start backwards. So this is a specific species of tick. So this is the genus species right here. So the genus and then the species. So this deer tick is what we know can transmit the bacteria that causes Lyme disease. So being able to accurately classify and identify this tick might be important. But if you work backwards, ticks belong to a larger group, a family, and there's different types of ticks. And then you back this up, you can see these are related. These are different orders. So mites and ticks here, and then some mites and some scorpions here. And then above order, so bigger, these are going to be different classes of arthropods, so all insects, and so flies and things in it, crustaceans, and then all arachnids belong here. So arachnids and insects are related because they're all arthropods. And so starting from the top, kingdom, ticks are animals. When you take animals, they're not chordates, they're not these flatworms, they're not these segmented or unsegmented worms. They're arthropods. And then so you keep breaking these down into smaller and smaller groups. So we use this today to identify all things. You can do the same thing for animals or plants or fungi. So this kingdom, phylum, class, order, family, genus, species, that is really important in biology. And so again, probably something you learned previously, but we're revisiting it in microbiology. So naming microorganisms still relies upon this binomial nomenclature that Linnaeus came up with, the genus and the species. And there is a certain way in which it's written or presented whenever you see it. So the genus is written first, and it's going to be capitalized, whereas the species comes second, and it's in lowercase. And when I say capitalized, just the first letter. So here's an example. This is a bacterial species, Enterococcus faecalis. So notice that's a capital E for the genus, but lowercase for the species. And the other thing for the genus and species, it should either be in italics like this one is, or underlined. And so that represents a scientific binomial nomenclature name. So in this case, Enterococcus faecalis. So that's how we write all binomial nomenclature names, whether it's Homo sapiens or Staphylococcus aureus, or in this case, Enterococcus faecalis. More recently to Whitaker, Woese, so this is another scientist, so Carl Woese, so that's his last name. He proposed another additional kind of unit of taxonomy, so bigger and broader than kingdoms. And so he called these domains. And so domains are based upon a new tool that we had in our toolbox for taxonomy that relied upon genetic information. And so the ability to kind of read that instruction manual that exists in all living things. And so specifically he focused on what are called ribosomal RNA, or rRNA. And when he looked at this across all living things, he noticed that there's kind of three major groups in terms of differences in these ribosomal RNA sequences. And so he proposed that we could organize all living things into three domains. And so they are listed here. Eukarya is one domain, bacteria is a second, and archaea is a third. And so those are the three domains. And so one other way of thinking about this is the two groups of prokaryotes, so prokaryotes include bacteria and archaea, are split into two different domains, because although they share some characteristics, that's what makes them prokaryotes, they also are different enough from one another that they can be placed into their own separate and unique domains. And that's indeed what Woese came up with, and that's what we accept today. So there's three domains of life based upon what Woese researched and proposed, and that's on genetic rRNA sequences that establish that. Then you can still use your same information beneath this. So what kingdoms are in eukaryotes? Well, we still have animals and plants and fungi and protists in here. And so those can be broken down further. And so that's indeed. So now we just have this additional layer, which is called domains. One other thing to realize, when Linnaeus first proposed this, it was a really important thing that he came up with, but he was kind of missing part of the puzzle, and that's what we call phylogeny. And so Linnaeus's goal was to classify and name things and catalog them just to describe all living things. We now know that the reason why things look similar or behave similarly to one another is because of what we call phylogenetic relationships. So just as you have your family tree, the more closely related you are to something, you're probably going to have more shared characteristics. So siblings might look more similar to one another than as you get to these distant cousins and second and third cousins and so on. So the same is true for the tree of life. And so what this shows are those same three domains, bacteria, archaea, and eukaryotes. And this is now looking at how similar those different subgroups are going to be to one another. So if we switch over to eukaryotes, what we can say is that animals are more closely related to fungi than animals are to plants because they're further away on these trees. And so not only do we want to put things in meaningful categories or groups and also have meaningful names, but we want to put those groups in a way that show shared evolutionary history. And so that's what was missing from Linnaeus. A lot of it that he did rely upon phylogenetic relationships, although he didn't understand that at the time. So a major goal in today's taxonomy is to understand the relatedness of organisms and put them into related groups. So that's an additional piece called phylogeny. When we take this information and apply it to bacteria, so another way of wording that is, how can we put bacteria in meaningful groups, or how can we look at their phylogenetic relationship? These are 5 different types of characteristics that can be used. So for example, we can look at the physical characteristics of a bacteria. So we actually look at some characteristic of it. So what does a bacterium look like under the microscope? That can be meaningful. What do bacteria look like when they grow on a petri dish? That's something. So that's called physical characteristics. Biochemical would be looking and asking questions about what is going on with their chemistry within the organism. Not all bacteria have the exact same biochemical reactions, and so we can test for the presence or absence of those. And that gives us a profile. And so now you can say not only, oh, this bacteria has a certain shape, and we'll talk about those shapes in just a second, but it has a certain biochemical profile as well. Serological are going to relate to blood tests and have to do with the generation of antibodies. We hear a lot about this because they have COVID-19 antibody tests as well. And so you can ask questions about the specific microorganism by what types of antibodies are present and what antibodies interact with specific microorganisms. I'm going to skip phage typing for just a second and move on to nucleic acids because I kind of already mentioned this. Nucleic acids is a fancy word for the type of macromolecule that describe either DNA or RNA. So there are two types of nucleic acids. There's DNA and there's RNA. And so if we think of those as instruction manuals present in every living thing, so while there's similar overlapping instructions across all life, there's shared biological pathways and information, there's some unique things as well. And so by looking at those unique things within the instruction manual or nucleic acid, we can learn about how certain bacteria might be more similar because they share more nucleic acid with their related counterparts as opposed to those that are more distant. And then last, this one is a little bit more complicated, and so I'll give it the brief explanation. Phage is short for bacteriophage, and so we'll see these in the future. Bacteriophage are viruses that can infect bacteria. And one thing that we'll see for viruses is that they infect very specific hosts and host cells. So when we say COVID-19, that's actually a disease. So SARS-CoV-2 virus infects humans. It can infect other species as well, but we know it can infect humans. It has specific cells that it does better in. So those cells of our respiratory tract. So as you get exposed to it, it can infect those cells lining your respiratory pathway. So viruses tend to be pretty specific. So these bacteriophage, or phage for short, they not only infect bacteria, but they would infect specific species of bacteria. So for example, Staphylococcus aureus, two words, genus species, Staphylococcus aureus. That's one bacterium. And so there are phage that would only infect staph. There are other phage that only infect strep. There are other phage that only infect E. coli. And so when we identify a new bacterium, we can get information about it. We can ask, what does it look like? Physical characteristics. What about its chemistry or its biochemistry? What about its serology within in response to within human blood? But we can now ask the question, what phage can infect it? And so all of these things give you a profile that allow us to better understand how we are going to group this newly identified bacterial species. Okay. So those are 5 types of characteristics that can be used. In terms of this course, physical characteristics are really important. We're going to talk a lot about those. Biochemical are also important. And then also nucleic acid. So number 1, 2, and 5 are probably the most important. But other than remembering that 3 and 4 are on the list, that's about all we're going to see them for. So as you develop answers to these different characteristics, you want to do the tests in an order or some logical progression that's going to allow you to determine what bacteria is present. So imagine a patient comes in and you have no idea what the bacterium is. There's a lot of different bacteria that are present on the planet, and a good number of those could cause disease. So how are you going to figure out which specific bacterium it is? So a dichotomous key is a series of tests that are done in sequence to determine what the organism can be. And this is not limited to bacteria only. This is really applied to identify whether it's birds or plants. You can use dichotomous keys, whatever you're looking at. But for obvious purposes, we're interested in bacteria. And so this dichotomous key is going to direct the user of the dichotomous key to what is the next appropriate test based on the answer you got previously. So this is the same dichotomous key that's presented in 2 ways. So I'm going to show you this side first. And so this is a little easier because it's kind of more in a graphical format as opposed to just words. And so you can kind of think of this as a flow chart or a concept map. And so the first question that you ask, is the bacteria gram-positive? And you can only get 2 outcomes from that. You can either say yes to that or no. And it's called a gram stain. We'll learn about that, but that's a test that can be done. Just as an aside, this yes has nothing coming off from it. Realize there's another dichotomous key that we could use that would allow us to go off in this direction and do additional testing, but this dichotomous key doesn't show that. So if it's not gram positive, so it's gram negative, Then this next one has to do with the shape. So are the cells themselves rod-shaped? It can be yes or no. And then so each of these yellow boxes is another question. And if you count the number of tests, so this is 1, 2, 3, 4, 5, 6. So these are 6 tests that can be done in sequence, and you've gone from I don't know what the bacterium is to this answer is the E of E. coli. So you've gotten down to the genus level. So remember, oh, this doesn't write very well. I apologize. That was supposed to be a G. So there's a hierarchy, and genus species is the lowest, most specific. And so in not too long a period of time, you can have the answer of what genus of bacteria it is by using this logical progression called a dichotomous key. So prokaryotes, we have mentioned at different times, they're small, simple, no nucleus, no internal organelles. They represent a huge range of microorganisms on the planet. It's really diverse, and this just shows some of those habitats that they can colonize and thrive within. The vast majority of prokaryotes, and more specifically bacteria that we are interested in are those that can colonize and grow on or in humans. And some of those, a subset of them, cause disease. So that obviously is the group that we're most interested in. But I do want you to realize there's a huge, and this is the vast majority of microbes are going to be growing and don't harm humans or other things that we know and love like our pets at all. So we definitely pay a lot of attention to these small, super small percent of bacteria that can colonize human and cause disease. So that's what we're going to talk about. So one way that we can study these microorganisms is obviously through the use of microscopes. Because we're fully online, you're kind of losing out a little bit on the ability to use microscopes. I trust that you have used them before in other classes. So we're going to touch upon this in lab at different times and do some demos and have some virtual microscopy that you do. But unless you have a microscope at home, you won't do any live in-person stuff. We're going to have you prepare some slides, but you won't be able to view them unless you have a microscope. So the point of a microscope is to increase the size of something, and so we call that magnification. There are 2 major types of microscopes that we will see images of throughout the course of the semester. And so light microscopes are probably what you're most familiar with. That's what you've used in intro bio class or in high school. And that has to do with using light waves. So a beam of light that passes through a specimen. And so that's what this individual, this researcher is doing here. And so this is a light microscope, a fairly fancier one, but it allows you to get images. There's also electron microscopes, which allow us to magnify something many, many more times than a light microscope would. This is a much more expensive instrument and requires a lot more technical skills to utilize and handling of specimens. So it's not available to kind of the masses. And so light microscopy is by far what's used most often because they're much less expensive and they don't require the same sort of upkeep and maintenance and technical skills. But the limitation is you can see the amount of magnification. Our total magnification is 1,000 times our human eye. That's pretty good. It allows us to see the bacteria. It will not allow us to see viruses, though. So most of the microbial groups, the microorganisms, we can see with light microscopes, but we wouldn't be able to see viruses. That would rely upon electron microscopes to see that. The last thing that we can talk about is on a light microscope, there are 2 sets of lenses that are going to help magnify. There are what are called the ocular lenses, and those are the eyepieces that you look through. And so each of those oculars or eyepieces has a lens in it that's going to magnify. And so that's usually, it's usually 10x. I just have to draw really slow for this to work. So if you were to use these by themselves and kind of pop it off and just look through it, it would basically be like a magnifying glass that makes something 10 times bigger. This is I probably shouldn't have used this picture because this is what's known as an inverted microscope, so it's a little bit different than the ones that you'd use in class. But there are these things right here that are called objective lenses that you can rotate or toggle between and choose which objective lens that you'd like to use. And they have different magnifications. But the way that you figure out how much the image is magnified is you take the 10x of the ocular and you multiply it by whatever the objective lens magnification is. And these range from as low as 4 to 5x on the lowest objective up to 100x on the highest. And so 10 times 100 gives you that 1,000 is how that works. I think that's all for microscopy. Even though we can magnify bacteria and see them with a light microscope, they're still really, really small and they're really hard to see. And so they are going to be small, colorless specks on a glass slide, super hard to see. And so the way that we can better visualize them is by staining them and adding color to them. And so that's the next thing we want to talk about. How can we take a bacteria? So say someone has a wound, a lesion on them that's infected, and we want to know, is that a staph infection or not? Well, you can take a sample of bacterium from the pus that's there and look at it under a microscope, but it's going to be hard to see the bacteria because, again, they're small and colorless. So what we first have to do is make what's called a bacterial smear. And so this is applying a small amount of bacteria on the surface of a glass slide so that it's a nice thin film that we can then see the bacteria after they're stained. The next thing is we let that air dry so that the bacteria kind of start to attach. But to make sure they're really firmly attached, we apply a heat source to the slide. This is called heat fixation. And so that has two purposes. It will kill the bacteria and also kind of cook them or adhere them to that glass slide so that as we're applying these liquids to stain, we're not going to have the bacteria lift off. Most bacteria have a charge to them. It's a negative charge. And so the dyes that we use so that it's attracted to the negative charge of the bacteria typically have a positive charge. And so we're going to talk about different types of stains, but most stains are going to have this positive charge so that it will interact and provide a color to those negatively charged bacteria. The first thing that we want to talk about here is two different types of staining procedures. So in microbiology, we can talk about most stains and say, oh, that's a simple stain, or we can say, oh, that's a differential stain. And so we want to understand what are those two different possibilities and why we'd want to choose one over the other. So a simple stain, as it implies, is the simplest sort of staining procedure. You would use just one simple dye, and it's going to be positively charged to interact with the negatively charged bacteria. And what you're going to get is all the bacteria, it doesn't matter what type of bacteria, they're just going to be stained one specific color. And what that will allow you to do is, regardless of the bacterial species you choose, you'll be able to make out their general size, their general shape, and something we haven't talked about yet, we'll see this in a little bit, it's called the arrangement of the bacteria. So size, shape, and arrangement. So that may be simple enough, and that may be all that you need to do. But oftentimes you want to learn something about the bacteria at the same time. So a differential stain is going to use not one dye, but rather two positively charged dyes. So that's one of the major differences, is one dye versus two dyes. And so in a differential stain, because there's two dyes, you don't use them at the same time. You use one stain first, that's called the primary stain. And then the second stain is going to be called either the secondary or the counter stain. And this differential stain procedure will give you the same information. You'll be able to see the size, the shape, and the arrangement of bacteria. But there's something else it gives you. Because two different dyes are used, most bacteria that are used, it's going to be able to answer a question about the bacteria above and beyond size, shape, and arrangement. So it's going to allow you to differentiate something about the bacteria. So for example, you can ask the question, is the bacteria gram positive or gram negative? And that tells us something about the cell wall or cell envelope structure. Or we can ask the question, does this bacteria make endospores? It either does or it doesn't, or do they have a waxy cell wall? These are our three examples of differential stains that we're going to see in the next several slides. So the Gram stain is differential because it has two dyes, and each bacteria that's put through it will end up with one of those two colors. So we'll talk about that in just a second. But each of those colors is going to be meaningful. I'm trying to remember which lecture it's in. I think it's the first one, so it's the history. We talked about Robert Koch, and in his lab they developed the Gram stain, and at that time we introduced the colors, either purple or pink. So those are the two end results, and each of those two colors means something about the bacteria. If you put an unknown bacteria through a Gram stain and it results in a purple stain, we conclude that's Gram positive. You put a different bacteria through and it stains pink, we say, oh, that means it's Gram negative. And so, yes, you see the size, shape, and arrangement, but you learn something above and beyond, and so something you can differentiate.